So this video is to show you how to go from DNA to mRNA to tRNA to amino acid using the DNA sequence. And so basically, the first thing you need to remember is that when you are going from DNA to mRNA, you are making an RNA sequence. So don't forget, with RNA, A pairs with U, and C pairs with G, just like normal, all right? And so what you would do first with mRNA is to basically just do the opposite. So thymine, remember we're going from DNA to mRNA, thymine would pair with adenine. Adenine would pair with, do not put thymine, because remember we're making an RNA sequence, so that's when you'd start using uracil. So U and then G, right? Now what you should notice automatically is AUG is a start codon. So this is when our ribosome would read our message and say start translating. So AUG, and we'd have G, C, G, A, G, G, C, G, G, C, A, G, C, U, G, and you'll notice my last here is UGA, which is a stop codon. All right, so I know it's a bit confusing because, sometimes we're using adenine and then uracil. Just don't forget, you are going to an RNA molecule. So you are, remember in the process of transcription, which is what this is here, going from DNA to mRNA is transcription. You are pairing, doing the complementary base pairing of these bases to your DNA strand. So that's the mRNA sequence. Now remember, tRNA has the anticodon or the opposite of your mRNA sequence, because remember, transfer or tRNA, it's what is going to be delivering our amino acids to our mRNA sequence, which is in our ribosome, to then form that chain of amino acids and ultimately our protein. So you would just do the opposite sequence again. And don't forget, we are working with RNA here. So this tRNA sequence is just the opposite of our mRNA sequence, okay? And what you'll notice is it's actually the same as the DNA, but instead of T's, you have U's. But you don't have to think of it that way if it's just easier to go from your mRNA to tRNA. So this is your anticodon sequence. So these are your tRNA, this is your transfer RNA bringing your amino acids. All right, so for the amino acid portion, we do need our codon chart. Now the part that you're gonna have to remember that's easy to get confused, and I wrote this at the top of that worksheet, your amino acid sequence is based on your mRNA, because remember the mRNA is what is coding for the amino acid. This is the translation part, okay? So the tRNA is just bringing the amino acid to the mRNA, but we have to read the mRNA to get our amino acid. So what that means is, okay, first let's start with our first set of codons, AUG. Go on over to our codon chart. AUG is right here, which is our start codon. It is MET. So I'm going to write MET. That's our first amino acid. Then we're going to have GCG. All right, so we got G, CG, that's ALA. Right, and so we're just gonna go on and on until we have all of these. So AGG is ARG. CGG is another ARG. And then we have CAG, just GLN. CUG is leucine. UUA, another leucine. UGG is tryptophan. And then finally, UGA is a stop codon, so you would just write stop. Okay, so essentially what you just did is you took a DNA sequence that you would see in someone's DNA. You then transcribed it. So transcription is you made a copy or that messenger RNA. Then a ribosome translated that mRNA sequence into an amino acid sequence or a polypeptide. Ultimately, this is just a protein. And that would be the translation portion. Okay. So you can expect to see this on the exam. You'll have the codon chart to look at. And please let me know if you have questions. I gave you the worksheet for a reason. I'll be happy to check it for you. There's also tons of YouTube videos that can help you. Just let me know.